was an undergrad, we actually had a lot of field trips. And in these field trips, we were looking at birds and we were also looking at small mammals. I wanted to go to graduate school, and I applied to about half dozen applications throughout the United States and Canada. And I applied primarily to work on bird evolution. My CV was grabbed by someone who is still at the Museum of Natural History, at the American Museum of Natural History in New York, and her name is Nancy Simmons. She grabbed my CV and she said, well, I know that you applied here to work with birds, but if you're willing to consider working on bats, I'm willing to support your application and you can come and work with me in the lab. And she gave me like a list of different projects that she was interested in. I had a couple of other opportunities. Actually, one of them was with birds, but I chose the one in New York City because I wanted to be in New York City. I wanted to be in a big city that was walkable, that there was public transit, and so I picked the M&H in New York City in the Upper West Side, and I picked my advisor on the basis of the public transport system. For the longest time, I think roughly since 2014 or thereabouts, I had become very interested in the olfactory receptors of bats. And olfactory receptors are really interesting because olfactory receptors are the single largest family of genes in the genome of humans. So I've become interested in the genetics of them because they evolve really quickly, because they expand, they duplicate, and they also contract. Some of the copies are lost, and because they seem to be roughly associated with the ecology. So starting around that time, 2013, 2014, we started analyzing what were the tissues inside the noses of bats. And I had this fantastic student, Laurel Yo, and Laurel was starting out her dissertation. And she was curious not only about the genes, but she was also curious about the actual structures that are inside the nose and how different they are from everything else. So by the time the pandemic came around, we had this unique data set that no one else in the world had that was focused on the genetics and the morphology of bat noses. Now, people like me, we're very interested in finding out like what is special about this rhinolophus bats, the bats that seem to be implicated in having this related viruses, right? And one of the things that is being found more and more in people who are specialized in that field of virology, and I think about that, is the concept of One Health. The concept of One Health is that our own human health and the health of the animals around us and plants around us is the same. Meaning, if we actually kind of go into a place and we destroy the forest, change the environment, have more interactions, enter into caves without any kind of protection, we do all of this huge human intervention without considering how are the plants and animals doing, how are they being restored, or instead everybody's stressed out, or instead now the bats don't have anything to eat, right? So that's the concept of One Health, is that the health is one. And I think this concept of One Health, the research on using this tool of One Health seems to show that these places where humans are very rapidly transforming landscapes and transforming them into either plantations or bare spots or development without any mitigation to make room for these species. These are the places where you actually have very high counts of viruses and very high risks. Obviously, it's not a fantasy that bats seem to have viruses that are relatively dangerous, right? But at the end of the day, we have created a world in which it's easy for these viruses to jump and to spread around the world. There is a very large gap, biological gap, that happens when a virus that circulates in wild populations in bats, for it to become a human pathogen. And that gap persists. We still don't know, we don't know how the virus evolved, what changes it underwent, or in which host, or how, we have like this darkness. In bats, even though none of the bats that we work on are suspected of harboring relatives of this virus, right? We can study whether or not there is expression of the receptors that bind to coronaviruses, such as this one, right? And that is the viral tropism, right? Is there a potential for the cells in the oropharyngeal cavity of the bat, which we have been studying in order to understand the symptoms of smell? Is there a potential for viruses of this kind to bind, enter, and replicate? Another aspect that we'll be working on, or we're asking ourselves, is how is different. So one of the problems with the human pathogen, right, is that if you think about it, if you think hard about it, the fact that the virus has such terrible consequences, particularly for the elderly or for people who are under particular kinds of treatment. It has huge consequences, bad illness, probability of death. But for other kinds of people that are younger, or women, or even, right, it actually doesn't seem to have as much of an effect, or the effects are, for the vast majority of patients, they're very mild. So that tells us something about the condition of the organism that the virus interacts with plays a very large role in the illness. That is called pathogenesis, right? And so one of the things that we can do in the bats is we can actually, again, without infecting anybody and without really looking at necessarily the exact bats involved, we can actually ask ourselves, is the genome of the bats, does the genome of the bats have all the elements the same as a human, or is it missing some parts, or does it have more additional parts in that interaction with the virus in terms of the immune response? Like if we wanted to create a model of the immune response of the bat, how would the model in the bat differ from the model in the human? We actually created this genomic model of the human response in the bat that tells us that bats really just, their immune response, although robust and capable of clearing viruses, it does not involve as much inflammation. It is an anti-inflammatory response. This is not a novel result. Other people have had these results, but we were able to confirm it with more than 37 species of bats. So right now we created a model that's just based on the genome, right? And as we know, like there is a big difference between having a genome and actually having an organism, right? Or even, right? So there's a big jump. There's levels of organization and things that need to happen, expression, regulation, and all of that. And I think the next frontier for us is going to be using cell lines and or transforming animal models like a mouse, for example, knocking in certain bat genes to have mechanistic observations of how these bat genes work, how they are actually different. I believe that the simplest path forward is going to be trying to use cell lines, bat cell lines, to test these ideas that we have about inflammation. I think characterizing the biology of bats is, in and of itself, is interesting. For a bat biologist, characterizing the biology of bats is interesting. But the biology of bats has become very relevant because, one of the interesting outcomes of SARS first, MERS, and SARS present, SARS-CoV-2, one of the outcomes of that is that these human pathogens that are related to bat circulating viruses, they tend to have really negative effects when they reach other species. We already know that in humans, getting treatment for the inflammatory response can be a good therapy in cases of COVID-19 infections, right? So all of this model that we have from the genome of bats could point in directions that we haven't tried yet. If we think just in utilitarian terms, bats do so many things for us. So there are estimates of billions of dollars in savings in just the state of Texas from insects. Now, if the bats were gone, as they have been, for example, in this region of the Northeast, this fungal pathogen, white nose fungus, that was introduced by people in 2006, has wiped out a huge number of bats. If the bats were gone from the state of Texas, it would be estimated that you would have to replace billions and billions of dollars and use them in insecticide. Instead of having the bats, we just have this natural pest control that is contributing to avoiding putting all of that pesticide into our bodies and into our land and into our water, and the bats are doing it for free. If we didn't have bats, we wouldn't have tequila, because there are several species of bats that are actually absolutely essential. We wouldn't have regeneration in tropical forests. If we didn't have bats, the chances of regenerating, growing back bare patches in tropical forests throughout the world, including Latin America, in Asia, and in Africa, would diminish, because bats are amazing at being seed dispersers in these tropical environments. So our entire world, even though we don't see bats very often, and most people may have an image of bats that's tied down with fantasies and scary fantasies, in reality, our very livelihood and our ability to persist into the future and have crops and have a clean environment that is not completely full of pesticides depends on maintaining populations of bats around the world.